Welcome to this lecture on the neurobiology of perception. Today, we're going to explore how our nervous system translates the physical world into electrical signals that our brain can understand. We will uncover the mathematical rules that govern our senses, from how we notice a change in brightness to how we detect the sharp edges of objects. By the end of this session, you will understand the fundamental principles of how we perceive the complex environment around us. Now that we know what we will cover, let us start with how we experience changes in our environment. Imagine sitting in a completely dark room and lighting a single candle. The change in brightness is huge and immediately noticeable. But if you light that exact same candle outside on a bright, sunny day, you probably would not even notice the extra light. Why does this happen? It is because our brain does not perceive the world in absolute terms. Instead, it measures relative changes. The way we perceive a new stimulus depends entirely on what is already there in the background. This relative scaling is a fundamental quirk of our nervous system. Building on this idea of relative perception, we must ask why our nervous system behaves this way. The answer lies in survival. Our sensory neurons operate using logarithmic firing rates, which compress sensory information. If our neurons fired in a purely linear way, a bright, sunny day would instantly max out our visual neurons, leaving us completely blind to any further changes or movements. By compressing the data, our brain can handle a massive range of intensities without overloading our delicate neural circuits. Engineers actually use this exact biological principle when designing audio compression algorithms and digital cameras to mimic human sight and hearing. Now that we understand the concept of sensory compression, let us look at the mathematics behind it. This brings us to Weber's law. Conceptually, this law states that as a background stimulus gets stronger, you need a much larger change in that stimulus to actually perceive a difference. The formula is delta I equals K times I. Here, delta I is the just noticeable difference, meaning the minimum change required for you to notice it. The variable I represents the initial background intensity. Finally, K is the Weber fraction, a constant that depends on the specific sense being measured. If the initial intensity I increases, the required change, delta I, must also increase proportionally for your brain to register it. With this formula in mind, let us see this with a concrete example. Suppose you are holding a light weight of 100 grams in your hand, and the Weber fraction for weight perception is 0.05. We want to find the minimum additional weight needed to notice a difference. We start with our standard formula, delta I equals K times I. Next, we substitute our known values, replacing K with 0.05 and I with 100. Multiplying these gives us a final answer of 5 grams. Physically, this means if you add only 4 grams, your nervous system will not register any change. You must add at least 5 grams for your sensory neurons to increase their firing rate enough for your brain to perceive it. Having seen how a light weight behaves, let us calculate the threshold for a much heavier weight. Now, imagine you are holding 1,000 grams, which is 1 full kilogram. We're dealing with the exact same sensory system, so the constant K remains 0.05. We want to see how the threshold changes when the baseline is 10 times larger. We again use the formula delta I equals K times I. Substituting our new values, we multiply 0.05 by 1,000. This gives a new just noticeable difference of 50 grams. Notice how the previous 5-gram change would now be completely invisible to your nervous system. Because the background weight is heavier, your neurons require a massive 50-gram increase to signal a noticeable change. Now that we have calculated these thresholds, let us visualize this relationship on a graph. What you are looking at is a plot of Weber's law. The horizontal axis represents the background stimulus intensity, labeled as I. The vertical axis represents the just noticeable difference, labeled as delta I. As you can see, the relationship is a perfectly straight line, starting from 0. The slope of this line is constant, and it represents the Weber fraction, k. This diagram beautifully illustrates that as the background intensity moves further to the right, the required change to notice a difference moves proportionally higher. It is a perfect visual summary of how our nervous system scales perception to avoid being overwhelmed. This understanding of sensory scaling naturally leads us to another fascinating trick of the nervous system. Have you ever noticed how the edges of objects look slightly enhanced or sharper than flat surfaces? This is not an accident of lighting. Imagine a group of friends trying to shout over each other in a crowded room. If one person shouts, the people right next to them might quiet down to let the main voice be heard. In your retina, neurons do something very similar called lateral inhibition. When one visual neuron gets excited by light, it actively suppresses its immediate neighbors. This creates a sharp contrast that highlights boundaries, sending only the most important edge data to the brain. To understand how lateral inhibition works physically, we must look at the structure of a neuron's receptive field. A receptive field is simply the specific region of sensory space where a stimulus will trigger that particular neuron. For retinal ganglion cells, these fields have a center surround organization. This means the center of the field responds to light in the exact opposite way as the surrounding ring. If light hitting the center excites the neuron, light hitting the surround will inhibit it. Without this architecture, our brains would just see a blurry, washed-out world with no distinct boundaries. This mechanism is so powerful that computer scientists copied it to create edge detection filters for artificial intelligence. Now that we know what a center surround field looks like, let us define it mathematically. Neuroscientists model this antagonism using the difference of Gaussians function. Conceptually, this formula subtracts a wide, shallow inhibitory curve from a narrow, tall excitatory curve. The formula calculates R of x, which is the total neural response at a specific spatial position x. The first term represents the excitatory center, where A sub c is the peak strength and sigma sub c is its width. The second term is the inhibitory surround, with its own peak strength, A sub s, and width, sigma sub s. The negative sign between them represents the inhibition. If the surround width increases, the inhibitory effect spreads out, changing how the neuron detects contrast. Let us apply this complex formula to a simple scenario. We want to find the firing rate when a pinpoint of light hits the exact center of the receptive field. So position x equals 0. The center strength is 100 spikes per second, and the surround strength is 20. Why do we choose x equals 0? Because it beautifully simplifies the math. When we plug 0 into the exponents, the numerators become 0. Since any number to the power of 0 is exactly 1, the complex exponential terms completely drop out of the equation. We are left with just the peak center strength minus the peak surround strength. 100 minus 20 gives a final answer of 80 spikes per second. Physically, this strong signal alerts the brain that a bright spot of light has been detected right in the center.
Having seen the response to a pinpoint of light, what happens if a broad, uniform light covers the entire receptive field? We are testing how the neuron responds to a flat surface with absolutely no edges. In a perfectly balanced neuron, the total area under the excitatory curve equals the total area under the inhibitory curve. Let us say the total excitation integrates to 100 units, and the total inhibition also integrates to 100 units. We simply subtract the total inhibition from the total excitation. 100 minus 100 gives a final answer of 0 spikes per second. This is a profound result. It means the neuron completely ignores uniform, flat light, proving that our eyes are designed to detect boundaries rather than absolute brightness.
To fully grasp this edge detection mechanism, let us look at the visual representation of the difference of Gaussians. This graph is often called the Mexican hat model. The horizontal axis represents the spatial position on the retina, and the vertical axis is the neural response, with a zero line in the middle. Notice the tall central peak pointing upwards? This is the excitatory center, where light causes rapid firing. On both sides of this peak, the curve dips below the zero line into negative territory. These dips represent the inhibitory surround, where light suppresses firing. This diagram perfectly illustrates how lateral inhibition filters out useless background information and highlights the edges of objects, creating a high-contrast map of the visual world. We have covered a lot of ground today, so let us summarize the most important ideas. First, we learned that perception is relative, not absolute. Our nervous system scales our perception based on the background environment, a principle defined by Weber's law. Second, this logarithmic compression is vital because it prevents our delicate neural circuits from being overwhelmed by massive changes in stimulus intensity. Finally, we explored how retinal neurons use center-surround receptive fields and lateral inhibition to filter out flat, uniform light and highlight the edges of objects. You now understand the fundamental biological and mathematical steps of how we perceive the complex world around us. Great job!